Good morning, everyone. Welcome. Good morning, good afternoon, good evening. Welcome to the second International Society for Vaccines Mini-Symposium series, and the very first focused on RSV. I am Dr. Nnenna Chime, the global clinical franchise head for RSV Older Adult Program with Sanofi. And we have an excellent lineup of speakers today. But before we get on with the presentations, I have a few announcements. We have the ISV webinar series, the next ISV webinar series on the 30th of April, 2025. Then the next. We also have the Next Generation Vaccinologist webinar series on the 8th of May, 2025. The annual Congress is coming up in Stellenbosch, South Africa, from the 28th of October to the 30th of October. And registrations, we're open for registrations and abstract submission, and we have a lot of opportunities for sponsorship and exhibits. And if you're not a member, please join. There are lots of benefits to joining ISV. And if you currently have your membership and it's up for renewal, please go ahead and do so. And finally, a very big thank you to our annual supporters and board members for their commitment and or financial support. We're on social media. If you'd like to keep following us, please join us on social media. We have various options. I'm going to give a very brief overview of the current RSV landscape before we jump into the presentations. So we've had a lot of advancements in RSV over the past few years. We currently have several products approved on the market. We have the vaccines, we have monoclonal antibodies for the pediatric population, and we have a very busy pipeline with multiple products in phase 1. We have monovalent, we have combination products, we have different modalities, right? We have the protein-based nucleic acid, and we have a few of them that have advanced to phase 2 and then phase 3. So things are really moving along. This is a very exciting space, and we're really happy to talk a bit more about it today. And on to the next speaker, we have Dr. Louis Bont, who is currently the professor at the University of Utrecht. He's had a lot of contributions to RSV. He's actually the co-founding chairman of RSVNet. And Dr. Louis Bont, please, can you go ahead?
Thank you so much, Nnenna. And welcome, everybody. It's a privilege to be here among so many distinguished speakers. I will share my screen, and no need to confirm unless you don't see my screen move. So I'm a pediatric infectious disease specialist working here in Utrecht, the Netherlands, and I devoted my professional life to fighting the consequences of RSV infection. And today I will talk to you about the burden of RSV. Most of my work is on the infant space, but also substantial part on adults. So these are my disclosures. Feel free to contact me if you would like to receive my presentation. So I would like to start this webinar with a patient, Francis. He was admitted to the intensive care unit here at the Wilhelmina Children's Hospital. After being born at gestational age 35 weeks, he went home after about 5 or 6 days after being born to meet his older sister. It was the middle of the winter season, and his sister had a runny nose. And she infected Francis, who got short of breath and very rapidly deteriorated, went to the ER, and the same day he was admitted for mechanical ventilation at our intensive care unit to survive, because his lungs, he was no longer able to survive without support of a ventilator. After 1 week, we could take out the tube and he was able to breathe again himself. A few days later, he went to a normal pediatric ward, and after 2.5 weeks of hospitalization, he was able to go home again. And still, up until today, he has recurrent episodes of wheeze following this RSV infection. And all of this is not just a single patient. Basically, this is the patient that is representative of the problem of RSV infection during early childhood. What I will talk to you about today is the burden during infancy and beyond. And I will talk specifically about low and middle income countries and older adults. So RSV infection causes 33 million disease episodes in children under 5, 3 million hospitalizations, 120,000 deaths, most of which occur in the first months of life. And this means that RSV is the second cause of death during the infant space following malaria. So it's really an important disease to fight from a mortality point of view as well as a morbidity point of view. When we look at RSV epidemiology, it's clear that all children are infected in the first 2 years of life. And following a rule of thumb, and we will be more precise in a second, 10% of those will require medical attention, 1% of those will require hospitalization, and there are a few high-risk populations, such as the premature child, as well as children with chronic lung disease, congenital heart disease, or Down syndrome. But it is important to realize that the vast majority of children who are hospitalized for RSV infection were previously healthy term babies. So if we really want to fight the consequences of RSV at the population level, we need to fight RSV in the otherwise healthy baby. So if we take this pyramid of disease, I will go through it from mild to severe disease. So if we start with mild disease, not even medically attended, so children who have an infection, but now not even go to a medical doctor. And we followed a large healthy birth cohort a few years ago, and a few months ago we published that if you look at those infections, then you can see that duration of symptoms is quite similar to the medically attended RSV infection. And disease severity is less than medically attended for obvious reasons, but it's still substantial, and it is following a similar course. And if you look at quality of life, again, you can see that there is a substantial impact, even the mildest form of RSV infection during the first year of life. And this is like the chunk of the iceberg that you have to realize. But then if you go to primary care, so not hospitalized, but children who require medical attention, we published a few months ago, again, a study that comprised several European countries. And what we found that RSV infection in the first year of life is found in 39% of children with upper respiratory tract illness at the primary care. Many of them require several visits, and many of them get a prescription for drugs such as antibiotics, bronchodilators, and systemic steroids. And what you can see here is that depending on the time after disease onset, there is substantial symptomatology, particularly in the first year of life, and some also seen beyond the first year of life. And you can see that 1 month later, most of the symptoms are gone, but cough, specifically cough, is good to realize that a substantial proportion of children after RSV infection will still have cough 14 days later. There's also something to realize when you counsel parents of an RSV infected child. So let's move on to the hospitalized population. We followed 10,000 children from birth onwards with several of our collaborators in Europe. And what you can see is a quite uniform pattern of any RSV infection, medically attended RSV infection, or hospitalization. And what you can see here is that 1 in 1,000 of the children require mechanical ventilation, or 5.5% of all hospitalized children. Probably this is the magic number. If there is any number from my entire presentation you would like to remember, is that of all healthy term babies, 1.8%, or 1 in 56 healthy term-born children, require hospitalization in the first year of life because of RSV infection. 14% require medical attention, and 26% will have an acute respiratory episode that we can demonstrate. So 1 in 56, please remember that. So because today we are also discussing prevention, it's also important to think that, to realize that RSV infection is not the acute episode only. There are other consequences of RSV infection. And I can't go into detail of all the potential complications or symptoms, but this is a good reference to look into that. In the first months of life, you can think of all-cause pneumonia or RSV-related mortality, and then beyond, you can also think of recurrent episodes of wheeze or asthma, which I will discuss in a second. But you can also think about RSV as in the causal pathway of bacterial pneumonia. And a few weeks ago, we published this paper where we explained that RSV infection is both biologically, but also epidemiologically linked to pneumococcus pneumonia. And what you can see is that some of these mechanisms that RSV causes are dependent on host interaction. Some are independent from the host interaction. It's really direct interaction between RSV and the bacteria. And then there is the complex relationship between RSV, the microbiome, and the risk of pneumococcal pneumonia. So what about wheeze and asthma? RSV is related to at least 3 wheezing phenotypes. First of all, RSV bronchiolitis itself is a wheezing disease. Second is preschool wheeze or infant wheeze, so the direct consequence or follow-up that we can see in children infected with RSV infection. And finally, asthma demonstrated by symptoms and use of asthma medication and lung function and everything that we all know that is related to asthma. And if we want to understand the interaction between RSV and asthma, we need to understand at least these 3 wheezing phenotypes. So this is one of the many studies that looked into the relationship between RSV and asthma. And in a cohort in the United States, the Rebel cohort, it was shown that at age 7, 48% had a diagnosis of asthma after being hospitalized for RSV infection, and 35% had active asthma. So really a demonstration of an epidemiological association. But obviously, and if I was able to explain this story well, then you realize that this is a chicken or egg dilemma. Does RSV cause wheeze and asthma, or are both the consequence of a predisposition or susceptibility? And what has helped me over the many years is that it is helpful not to see this as an either or an exclusive hypothesis, but rather both hypotheses are true, and we need to define to what extent both hypotheses are true. So I'm going to show you evidence of both hypotheses. So the first one is a study that we did with several collaborators in the world that was led by Steve Brunwasser, who looked into genetic confounding when looking into the relationship between RSV and asthma or wheeze. And the idea was that if you control for genetic confounding, and there is a genetic component, then you would assume that the association decreases. And that is exactly what we found, showing that if you control for genetic confounding, the association is substantially lower, showing that there is a predisposition in place here. That is not just RSV as a causal factor, but there is really predisposition, and there is more in the genetic space that points to this association. But there is also evidence of a causal pathway. And this is a study that we did years ago in healthy children born at gestational age 33 to 35 weeks, a placebo-controlled trial with palivizumab, and we looked into the parent-reported wheeze. And what we found was that palivizumab not only prevented RSV infection, but it also prevented more than 50% of all-cause wheeze in the first year of life, and not just during the first winter season, but also long after RSV had left the country and in the middle of the summer, showing that RSV, at least in this trial, causes wheeze. We went on and followed these children up to the age of 6, and then there was still a relationship with symptoms, but not so much with lung function, making us doubt to what extent this association is actually persistent or transient during preschool years. And then there is this observational study from Tina Harter's group, the INSPIRE birth cohort, where they looked into the relationship between RSV infection and asthma, and not just severe RSV infection, but any RSV infection, even including serologically proven RSV infection. And they concluded, based on these data, where you see the relationship between RSV and asthma, that seems to decrease over time with age. The x-axis here is age, but that 15% of 5-year current asthma cases could be prevented by avoiding RSV infection during infancy. To definitely establish causality, we need interventions, which we have. And I showed you there is an age dependency here. So evidence that we are in a very interesting time where we can disentangle cause and consequences of this intriguing association. So the next topic I would like to discuss with you is the global incidence rate of RSV infection in children under 5 globally. And what you can see here is that the incidence of RSV infection and severe RSV infection is highest in the low and middle income countries and lower in the high income countries. And this is something that you recognize from every other disease or many other diseases, but also for RSV, there is an unequal distribution that you can see here in a review paper by Nathalie Mazur recently. So if we look at RSV in Gavi eligible countries, so the poorest countries in the world, we studied with RSV molecular testing the incidence or the proportion of RSV positivity among children that were life-threateningly ill, so at the intensive care units at in these 10 countries. And although some countries like Nepal had a lot of children included and others had lower numbers included, there is a very, very persistent and predictable pattern here. 30% of all children who need support to survive are RSV positive when they have respiratory symptoms. So it's 30% of all children under 2 with respiratory symptoms who have a life-threatening disease are RSV positive. And this is the first proof that also in those countries where people often neglect RSV because they never diagnose RSV, RSV is still a major cause of disease, confirming data from the epi field that were more dependent on modeling that RSV is a serious disease in the poorest countries in the world. And if we look at age distribution of those very sick children, then we analyzed, and this is we have confirmed this in other studies now, that if we look at our global mortality registry, that we can see that the majority of children who die from RSV do so in the first 6 months of life, which is really important because that opens the opportunity for RSV prevention. So before I go to the next slide, I would like to ask you to take your cell phone in hand, because later on I'm going to ask you to use it. So I will move on with my presentation, because I would like to show you that in low and middle income countries, it's not just a lot of disease and mortality, but many of the children who die never make it to the hospital. So if we look at several studies that were funded by the Bill and Melinda Gates Foundation, you can see that the majority of children who die do so before they reach the hospital. And I think this is an important message, that unless you look for it, you don't see RSV disease. So this is a summarizing slide that I hope you appreciate of RSV burden in children under 5 in the world. So 31% of pneumonia in these children is caused by RSV. These numbers I told you already. And I think you appreciate that the vast majority, almost all children who die from RSV do so in the low and middle income countries. Most of them do not have access to intensive care, so they die because they don't have access to the care that I showed in the first child in my own hospital. They don't have access to pediatric intensive care. There's a huge amount of money involved, and what I showed you is that there is a lot of morbidity and mortality out of hospital, and that age at death is very young. Oh, again, a positive signal that we may do something well with maternal vaccination or monoclonal antibody prevention. So the last topic is RSV in the elderly. This is the most important study in this field, published almost 20 years ago now, where Enfalsi looked at elderly patients with COPD or heart disease in the community, or among hospitalized elderly with pneumonia. And what you can see among elderly is that there is RSV infection. 46 out of 608 developed an RSV infection, similar or even more than influenza, but very few required hospitalization. So from the population perspective, severe RSV infection is relatively rare. If you're a healthy older adult, the odds of being hospitalized is really not so bad. But when you take the population perspective of elderly with COPD or heart disease, then the incidence of RSV infection is not so much different, but the proportion of those people who require hospitalization suddenly increases massively. So among high-risk adults, RSV is a bad disease. And taking the hospital perspective, if we look among hospitalized people, then 142 out of 1,388, so more than 10% of all pneumonia hospitalizations were RSV positive. And now we start to see mortality. So 8% of those with RSV infection actually died, showing that RSV is a disease that is frequent, and dependent on your perspective, it's a bad disease. Many other studies followed, and this is a slide that actually summarizes that: the community perspective, the medically attended respiratory or the hospitalized perspective. And then if we look at incidence and outcome setting, then you can actually see that the incidence is between 3 and 7%, as I just showed you. That medical attention is relatively rare. Only 1% require medical attention if we take the community perspective. If we look at primary care physicians, then the incidence is not very different. But now we start to see hospitalization. About 1% of those require hospitalization. Mortality is still low. And then again, if we take the hospital perspective, then the incidence is again not so different, but now we start to see mortality. And I think this really boils down to the conclusion that RSV infection is frequent and dependent on the perspective, it is severe or even more mortal. And what are the complications? I thought this was a nice review article where in the upper graphs it was shown that there is a group of adults that are at risk for bad disease, in particular older people with heart or lung disease, that there are early complications of RSV infection, include in particular exacerbations of the underlying disease, but also bacterial co-infection and intensive care unit admission, cardiac complications, and then there are late complications that have to do with long-term loss of function, fatigue, etc., that are quite intuitive. But again, a disease that you want to prevent. And you will hear later on that we can prevent these diseases also in the older adults. So before I give you time for some questions, I showed you that RSV infection during infancy is substantial, 1.8% of all healthy children, that there are long-term consequences of which we are not sure to what extent these are causal. I showed you the low and middle income perspective. About 30% of all bad respiratory disease around the world is caused by RSV. And I showed you that older adults are also susceptible, in particular if they have comorbidities, and that there are complications. So I told you a lot. Some of you are RSV experts. I recognize your names in the list, but some of you feel you would like to know more about RSV or remember what I told you. So for that, we developed a chatbot that is really performing well. It was developed independent from any company by a non-profit organization called RSVNet. You can use it on your WhatsApp. So if you go to the RSVNet website, you can see it. But also, if you take your cell phone now, please use your camera and look at this QR code. And please scan it. I will take 20 seconds to talk you through it, and assuming that you're using it. And you can ask any question that you want to know about RSV in any language that is available around the world, and I am sure that you get the right answer. Many people use it now. The World Health Organization has complimented us that it really works well, and also companies and the Bill and Melinda Gates Foundation and Center of Disease Control. So I think it is fair to assume that this chatbot is working well. These are the people that I would like to acknowledge: my colleagues at Yale, with whom I do a lot of my EPI work, the RSVNet Foundation, close friends of mine, and at the right you see all the people that do the work that I was allowed to present today. Thank you very much.
Excellent. Thank you very much, Dr. Bont. That was a very insightful presentation. I don't see any questions in the Q&A. Do we have any attendees with any questions? You can raise your hand, right, if you have any questions, and we can let you come off mute. And I think while we're waiting to see if the audience will have any questions, I have a question for you. So something struck me during your presentation. You said, if you don't, unless you look for it, you won't find it, right, with RSV, when you were talking about low to middle income countries. And I'm just wondering, we know that there's been a lot of work done with RSV, but there's also some growing interest in HMPV. So do you have any thoughts on, right, just based off of that concept of if you don't look for it, you won't find it? What do you think about our current understanding of the burden of disease of HMPV in pediatrics and in adults?
So HMPV is virologically from the same family. It causes similar disease in children and in adults, probably less. So the burden in infants is less, but it could be considered the next kid to think of. But I have a little bit of a bias, because I think until we have started to prevent RSV disease around the world, we need to focus on that problem before we go to the next pathogen. What I've seen too often in vaccine development is that we develop those vaccines and immunizations for part of the world, and not taking care of the rest of the world. So I honestly feel that it is our responsibility here to address that together, including everybody here. I hope you agree that that is also part of our responsibility. But having said all of that, HMPV is an important disease to consider when we're seeing a child with pneumonia. Thank you.
Okay, thank you very much. Do we have any questions at this time? Okay, well, thank you very much, Dr. Bont. I think we'll go on to the next speaker now. And we have a Dr. Peter Kwong. He's had a lot of contributions to RSV, structural biology of RSV, and he's currently a professor at Columbia University. So Dr. Kwong.
Thank you so much for that introduction. Let me share and presentation. Here it comes. I put that in presentation mode. Sorry. There. Does that look okay from your screen then? So I'm absolutely delighted to be here today to speak about technology. We had a wonderful introduction to RSV itself, but I'm going to be speaking about Prefusion-F stability and vaccine efficacy, tertiary-based vaccines for RSV, HMPV, and PIVs. I'm going to be speaking about 3 topics today. First, for the technology development, I'm going to say a little bit about HIV, which is where we developed technology and apply it to RSV for structure-based vaccines. Then I'm going to go into HMPV, the second one going forwards, and then even further out to HPVs and other paramyxed viruses, even more diverse for structure-based vaccine design. First, I just want to state for the record that you do not need structure for vaccine development. You can go straight from virus to vaccine by, following Willis Hillman's isolate, inactivate, inject, and that works for many different pathogens. However, there are a number of pathogens that it doesn't work for, and as a structural biologist, many of these were under investigation from Don Wiley and Ian Wilson's hemagglutinin that was solved in 1981, or Michael Rossman's rhinovirus. And then I did the G120 CD4 from HIV with Wayne Hendrickson. And so with all the structural information, the question that arose based upon, in part, the structure-based development of drugs, could you use the same atomic level design in vaccines? And so I do want to give a little bit of a definition here, which is there's structure-assisted vaccine design, where you can use structure to assist the development of vaccines. And in influenza, we've had the structure from Ian of the hemagglutinin, and that information is used in the vaccine, but not necessarily directly, and that people just consider it when they're developing where things are on the surface. But it's not used in exactly the immunogen design process, where structure-based vaccine development, where you use immunogen design based directly on atomic level structure, is a little bit different, and that's more akin to what one does for structure-based drug design. So in order to answer this question, I'm going to go back about 25 years when the Nnenna Vaccine Research Center was put together by Gary Nabel. It's a really interesting plant that did basic science all the way to clinical trials, and Gary Nabel, shown here, pulled about 10 of us PIs into that had all sorts of different forms of vaccinology expertise. Mine at the time was structural biology, and people were like, oh, why do you need a structural biologist in the vaccine area? And Gary partially explained this in that it's not necessarily clear that when you're trying to solve an important problem that you actually have hypotheses about, but rather you should collect data on important regions that you know from other regions why that should be important. And so I was trying to generate data on envelope structures, antibodies, and design of vaccines. And so the initial part of that led to, you can see maybe the first 10 years of the section, lots of conceptual breakthroughs, including things like conformational masking, where we found that the correlation between a neutralizing potency and receptor antibody receptors revealed a receptor binding site conformational masking mechanism of a neutralization escape, which allowed HIV to maintain receptor binding simultaneously to resist neutralization. HIV envelope is highly glycosylated, and by analyzing patterns of HIV escape, we came up with a glycan shield that prevents selected changes in glycan packing, and that allows escape. There are reproducible antibodies that came up as well, and we came up with ideas for how certain regions of the envelope were immunodominant, especially those that are bound by lots of different antibodies. Showed a site of vulnerability where there's certain broadly neutralizing antibodies that could bind many, many different diverse strains of HIV by overlapping the receptor binding surfaces. And in order to do that study, which was from 2007, we actually had to stabilize the G120 for structural analysis. And we did so by cavity filling mutations. You can see we introduced tryptophan here that stabilized this cavity, and also the disulfide itself, which was designed. So you can have cavity filling or disulfide stabilization that actually occur. When we did this, we realized that the stabilized molecules could be played in structure-based elicitation. And what that meant was that there was an epitope down here for CD4-induced antibodies, highly conserved, all the way from HMV1 all the way to HMV2. By putting in stabilization mutations, we could form this epitope for the CD4-induced antibodies. And if you look for stabilized G120s, only partially stabilized or further stabilized, this paper by Bont, she was able to see by immunization that if they're only partially stabilized, you don't get much of the CD4I antibodies that neutralize all the way to HMV2. But if you stabilize even further, you can get a lot of the CD4I antibodies induced. And so that showed basically how conformational stabilization of the envelope G120 could elicit particular antibodies. Unfortunately, those antibodies weren't able to produce certain types of HIV which were resistant, and so we need to pick a better antibody epitope. Nonetheless, that showed us how antibody capture conformations could be used in vaccine design, in part because one big trick was needed in order to do the actual structure-based vaccine design. We knew that antibodies could recognize conformation, and we knew conformation is very important in terms of vaccine development. But how to couple recognizing antibody and conformation they recognize going forwards? And so we figured out how to transfer conformation-specific information from the antibody to the antigen in this HMV paper, where we had one last part of HMV envelope, the B1B2 domain, didn't know its structure. We knew an antibody PG9 recognized it. So Jason McClellan actually took the PG9 antibody, inserted a cleavage site between the FAB and the FC. Tunching Joe said, hey, put that in a column and you can actually flow lots of things over it. And then Panzer actually did the experiment where she flowed over different forms of V1, V2 that were scaffolded, was able to get complexes. The ones that complex, I think only about 5% of the protein was actually in the right form that could be bound by antibody PG9. She cut this off with a protease site. So you have the FAB region with the antigen now in its right conformation, and she get the structure of that, which was done in this paper published in 2011. So the early stages for the development of the structure-based vaccine design technology, we showed that there are a lot of conceptual breakthroughs needed, such as conformational stabilization. And then we also showed how to use antibodies to capture susceptible states of a viral antigen. So let me see. I just want to do a pointer options. Let me make that to a laser pointer here. So this is my lab at the time, around 2010, 2009, and Jason McClellan said, hey, you've been working in HIV. Wouldn't it be really helpful to test those ideas with other viral pathogens? And so the one that was chosen, in part for the reasons that you just heard from Louise, is that respiratory syncytial virus would be the target. It's the second leading killer of children, of babies in this critical period to malaria. Greatest cause of hospitalization. Importantly, Barney Graham, who was chief of viral pathogenesis and also of clinical studies at the time at the VRC, had spent about 20 years before he came to the VRC to work in this. So we had someone who was an expert that could help us in neutralization studies. We decided to focus on the F glycoprotein because that changed shape, and also because it's analogous to the HV envelope G120 GP41 that was the viral entry machine for HIV. And so just like HIV, it's a class 1 fusion protein where the F glycoprotein binds receptor, causes conformational change, then induces post-fusion conformation along with a concomitant merger of the viral membrane with the target cell membrane entry of the virus. Barney Graham and I showed that if you look for prefusion-specific antibodies, after pre-F absorption, absorbed all the neutralizing activity throughout all age groups, whereas if you absorb a post-fusion, you were left with a lot of neutralizing activity was remaining. And so we knew that prefusion-specific antibodies were really the trick. And Jason McClellan found one D25 by looking at the patent literature. did the HMV3 cleavage trick there. Then followed overcome with forms of RSV that were not stabilized at all. This is wild type for the ectodomain. Cut those off and then had the FAB bound to the stabilized, the antibody recognized conformation, and could get crystals of that, which then could determine the structures of that. And once we had the atomic level structure of the prefusion form, and the post-fusion form that Jason had determined earlier, we could examine the protomer structure that's shown in the middle diagrams. And you can see enormous change between the pre-fusion conformation versus the post, but we had now atomic level detail. Since we had the atomic level detail, just like with the H3 envelope could stabilize with disulfides or cavity-filling mutations, here is one example of two cysteines at 155 or 290 that are very close and can form a disulfide bond if you mutate them into cysteines. And as cysteines, you can see that locks the prefusion conformation on the left and prevents the composition change to the post-fusion conformation on the right-hand side. So I did some immunization with these with Marnie. This is macaque data where you immunized at 0 and 4. And you can see for post-fusion, the black bars would raise the neutralization titers to very close to the protective threshold going forward, which is why so many companies tried post-fusion type molecules because they were almost there. But you can see that the pre-fusion disulfide stabilized in yellow, where the disulfide plus cavity filling in green gave higher titers. This is a log scale, so at the highest it was about 80-fold on average higher than the protective threshold versus about the same that you would actually see here going forwards. And so that led to a structure-based paradigm for vaccine development where we would take the most potent antibodies from infection, use those to stabilize the antibody susceptible conformation of the viral entry machine. Then through a 4-part cycle design, property, structure, and immunogenicity, we could optimize that. And that's how we came up with GSCAV1, which was considered the run-up to breakthrough of the year in 2013 when we published that. Because I was at the VRC, we could actually do manufacturing. And so the vaccine pilot plant headed by Dick Schwartz started making cell lines for DSCAV1. And at this stage here, I think we got to about 200 mg per liter. So it was looking really good. Ultimately, the productivity yielded over 1 gram per liter. The clinical trial for this GMP material showed that 150 micrograms of GSCAV1 with alum would increase the titers about 20-fold. And if you had no adjuvant at all, the titers would go up about 15-fold. So that looked pretty good. So that would lead to about 5 months of protection when added on going forwards. If you're looking for 6 months of protections for infants when their lungs are developing. So ultimately, this did go forwards to a clinical trial with GSK when they saw our initial results here going forwards. Then in 2022, phase 3 trial looked amazing with 82.6% reduction and 94% reduction of severe RSV. And that led to the FDA approving Arexa in 2023. All are good, but if we use exactly the same antigen, GSCAV1, we started working with Moderna and Merck to try to put this and deliver by mRNA. Unfortunately, that failed. So if you actually take just the disulfide stabilized one that works as a soluble ectodomain, it does not work very well. Just about 2-fold higher titers increase after its immunization as delivered by mRNA. What we did, though, is we could optimize using this 4-part cycle design actually compared structure and immunogenicity. And you're going to see this iterative optimization form later on for some of the other ones going for the HMPV. But since this is published, this reference by Gordon Joyce, we ultimately led to molecules that were 20-fold increase in stability and 4-fold increase in immunogenicity. And when Moderna tried those more stabilized double disulfide fixed molecules, you can see the increase in the young was really quite high, about 15-fold on average and over 10-fold for the elderly. And so just to summarize this part, we used the prefusion-specific D25 antibody to capture RSV and its antibody susceptible conformation, and that led to the GSCAV1 variant. GSCAV1 stabilized is licensed by GSK, approved, and now showed as Arexa. The second generation version of the conformation stabilization with the double disulfide succeeded in Moderna's phase 3 trials, and it's been approved by FDA, and so does RSV. So all this work was done at the NIH, and about 40 of us maxed out from the GSK and Moderna licensing and patent royalties as of last year. And so that completes the first part, the background, showing how some of the early HIV stuff could be moved over to RSV, and that led to the development of really interesting vaccines there. So I'm going to follow the second and third parts of the talk moving forward with HMPV, showing the same technology work there, as well as very much more diverse pathogens. And so for HMPV, second leading cause, lots of significant hospitalization. What's been published is the equivalent of a sort of DSCAV1, where we showed that you could have stabilizing disulfides and cavity-filling mutations. And in fact, Guillaume Stewart-Jones was able to find a V3b that gave really quite, they looked pretty good in terms of its yield. Well, actually, it wasn't that great in terms of its yield. It's only about 0.2 micrograms per 0.2 mg per liter, so it's relatively poor yield. But we could also get a post-fusion form, which actually get disulfides there as well. And we asked, if you take disulfide stabilized prefusion, disulfide stable post-fusion, were the titers. And what's interesting is they both increased over the background. And actually, the highest titers we had was the post-fusion disulfide stabilized molecule. So when we pulled all the data together, we could see that stabilization with disulfides, not necessarily into pre or post, actually did increase immunogenicity in mice that are going forwards, although they weren't enormous changes, but each disulfide apparently did help. And so we've asked the question, well, we had 2 disulfides for HMPV. What happens if we go further? One of the big problems here with this HMPV, the more disulfides you put in, oftentimes your yield goes down. So we had to recover the yield, and Leo did a very interesting trick by altering the linkage region between F2 to F1, and found that the productivity could increase quite a lot with these going forwards, and actually partially stabilized. So instead of having 0.2 mg per liter, you could see that some of the yields went quite a bit higher, and we had an interesting quadrant of MPE8. We could recover the binding of that, where the original V3 didn't actually bind that. And so we had additional disulfides to stabilize HMPV-F here going forwards. And then we decided to make these triple disulfide versions that are shown here, with also had decent immunogenicity and decent yield, some up to now almost 40-fold higher yield going forwards than the initial V3 molecule. Show the cryo-EM here. It shows that the disulfides all did form. And here's the critical immunogenicity. Whereas the DSCAV1 form of HMPV, or the double disulfide form, or even the post-fusion, give titers that are between 100 and 1,000. Now, when you start going to the double disulfide versions, the titers average about 10,000, and so they're considerably higher. And so, just to summarize this portion, the disulfide stabilization of either the prefusion or post-fusion HMPV-F induced higher neutralizing titers. The multiply disulfide stabilization HMPV yielded the highest titers. And then in mice, the triple disulfide version gave titers of over 11,000. So at least in our hands, more disulfide is better. And there's lots of questions as to why that's true. Is it merely the stabilization that is into conformation, or is the fact that if you stabilize these longer, they can hang around the germinal centers, give higher response, et cetera, et cetera. So many different reasons why this might prove. So this is just observational of what we see. And finally, going to the last part of the talk, which is you saw how we did HMV to RSV, then we went to HMPV. But what about, there's many other pathogens with the paramyxovirus family that also leads to human diseases. And so tackled these with Prabhanshu Tripathi, a talented postdoc, and here is my lab at the NIH. And so, as shown here, many paramyxoviruses induce significant disease burdens. Is there a general way to stabilize all these F-like proteins in this prefusion shape that looks a bit more immunogenic and easier able to get neutralizing titers? So there were initially 6 different disulfides that Guillaume Stewart-Jones was able to figure out as ways to stabilize PIV3. And so we decided to take the initial work that Guillaume Stewart-Jones did on PIV3, which led to decent titers, and so to see what is the pairwise identity. Could we use this disulfide stabilization in a general way to stabilize not only the paramyxoviruses that I'm going to show you about, but potentially all the way out to others that are quite further, like Nipah or others going forwards, mumps, for example. And so here I'm just showing you for the PIV 1 through 4 that there's quite good homology, and we could see different disulfides that we could work out. we decided first to transfer this the disulfides between PIV3 and HPIV1. And the reason we did that is because they're the closest within the phylogenetic tree with 45% identity. Now, when we did this, we said, okay, how do the disulfide yields, how does that affect the yield? The other important thing was AlphaFold tolerance. So we actually have to do this thing experimentally. And we saw from AlphaFold 2 that it said that all the disulfides would form with PIV1. AlphaFold 3 said that only 2 would form. And the answer, when we actually did this experimentally, was that 3 formed. And so they did get this one right, but AlphaFold 2 got this one wrong, and none of them got, well, they guessed these other ones. So here's the disulfide stabilization that's shown here going forward. So you can see that 3 of the disulfides did seem to work. We have to search for one of them, but all 3 induced higher immunogenicity. What about doing the same trick between PIV3 and PIV2? Could we transfer these over going forward? The sequence identity here is lower, only 26%. And the answer is that when we did this, we found 2 with really quite good disulfides that matched. They all formed PIV3, but when PIV2, 2 formed. We'll get the structures of both of those, confirmed the formation of disulfides, and then both of them showed a log increase over the post-fusion form of immunogenicity. One's a little bit better, 2, almost 3 logs higher. And finally, what about PIV3 all the way out to PIV4? And this is about 28% identity going forward. So the answer is we found 2 that also looked really quite good at 197%. When we looked at those structures and looked at their immunogenicity, they're also about a log and a half higher versus the post-fusion form. So basically, it's looking like it's working for the homology transfers out to PIVs 1, 2, 3, and 4 from the initial stuff for PIV3. But could we even go further? Could we take the pyramidal 1 and 3 information and go to the beta virus or the HMPV virus or some of these even further out? And so here's the homology stabilization. You can see that the regions that were chosen are actually in somewhat conserved regions, although many changes occur in these regions as well. And the bottom line is that we did find the PIV3 homology disulfide stabilized molecules did stabilize the Moderator virus, in this case, the stepper virus. We got the structure there at 3.5 with this particular disulfide. Mumps had 2 different disulfide, a Nipah virus with 162 to 168 disulfide. And so bottom line, to summarize this part, we identified 6 prefusion stabilized disulfide HMPV3F. The homology design showed prefusion stabilized F for PIV1, 2, and 4, and each of these showed at least 10-fold higher immunogenicity versus the post-fusion form. And finally, the homology design disulfides also work for even more diverse pure mix of viruses going forwards. What is an overall summary here to my talk? Just finishing up. The prefusion stabilized DSCF1 yields high titers as a protein. We could transfer the information over. An additional stabilization was needed for mRNA, but also led to a successful vaccine. In terms of HMPV, disulfide bonds appear to increase immunogenicity both in pre and post. And if you just kept adding them more on, we're up to 3 now, and that did appear to increase the immunogenicity HMPV-F. Is it just stability? Is the prefusion conformation fixation? Is it exactly why this goes up? Is still an interesting immunological point, the ZAC mechanism. And finally, we could transfer homology by disulfides from PIV3 all the ways to PIV1, 2, and 4, as well as the even more diverse mix of viruses. So just would like to thank a lot of people for this. Initially, Barney and Jason did amazing work in 2013 getting the initial DSCF1, and here we're all really happy about that initial paper that was going off. I led a large team at the NIH before I left there at the end of 2013, and here's about half this group is my team. The other half of the group is the people that actually made vaccines on the 4th floor and did a lot of different work there, as well as some other structural people that were there as well. And finally, I now have a small team at Columbia University. We're working on therapeutic antibody-based vaccines, and we're moving forwards in that context. So any questions, I'm happy to take. Thank you.
Excellent. Well, thank you very much, Dr. Kwong. I don't see any questions in the Q&A. And just a reminder, everyone, we're taking questions at the end of each presentation. So if you have any questions, please type it in the Q&A. And if you want to ask any question, you can raise your hand and we'll let you talk. And Dr. Kwong, while we're waiting to see if there's anyone in the audience that has a question, I have a question for you. So, well, thank you for all the work you've done with RSV and with the other viruses as well. Very fascinating and extremely helpful. You talked about the difference, right, in immunogenicity when you had the DSCAV1 with the protein and then tried it with the mRNA platform. It required more, right, the disulfide stabilization, right, for you to actually get the similar immunogenicity response. Do you have any explanation as to why that is the case? That's one. And two, do you think that that's also going to translate to, right, HMPV and how that is expressed? Because you didn't say anything about the difference between protein versus mRNA with HMPV.
So thanks for those interesting questions. Exactly the differences between the format of the protein used going forward versus the actual effect during the vaccination. Clear as you saw, and you summarized very nicely that got a great vaccine with DSCAV1 that was stabilized, but we always knew that DSCAV1 form of the protein wasn't fully stabilized. That is, if you incubated at 37 degrees, it had a decent half-life and would start converting or falling apart or doing other things to it. And so that's why we had gone to the second generation version in 2013 with double disulfides, and those were much more stable, 20-fold more stable, and going forward to heat and activation, and so much more stable. It turns out that it's likely that stability correlates going forwards for mRNA vaccines. Why that's true is not entirely clear, but I do think it's part because when these molecules presented in the germinal centers, it's not like they're necessarily presented for just a few minutes. Sometimes it can be days or even weeks that the germinal center takes these, develops good antibodies, and really pushes them forwards. And so the longer you can have a immunogen that has this right conformation fully in the germinal center, teaching antibody recognition of that conformation, I think the higher responses you're going to get. The issue that's going forward that's very complicated, however, is that the results that I'm showing you with the monkey or the mouse immunizations are with the naive animals. And what's very, very different is that you have, at least in the elderly or in many cases, a pre-existing response that the immunogen alters. And so that response is not so easy to look at because there's not necessarily a good pre-existing immune model for that moving forward. And so that's why in part we're like, okay, this is what we saw experimentally. And then your second part of the question is, well, would that be true for others going forward? And I already showed you that at least in the model with immunization with HMPV, the disulfides were helpful. But what does that mean in the context of the pre-existing model? I think very difficult to predict. I think you're just going to have to do immunization studies, the human clinical studies to really understand that. But because of the problems that we saw with conferential stabilization, not really being fully stabilized and failing in the initial DSCAV1 mRNA trials, we thought that we would at least design with our technologies many different disulfide versions. So make them public, available to the field. And then people could try things with mRNA, which is a much lower bar to enter in clinical trials and to see, does that work going forwards? So it could be that you could figure this out by some analysis of an interesting animal study. But if you're able to do mRNA immunization go into humans, I think you're going to get really interesting results. And that's why we made this not only for HMPV, as you've shown, but also for the PIVs 1, 2, 3, and 4. And ultimately, I think it's going to be experimental medicine question. What's going to be the best vaccine going forwards? But my part as a biologist is to show that we can actually have the technology to stabilize these molecules so we can actually test those. So does that answer your question?
Oh, yes, yes. Thank you. Thank you very much. Very insightful. And I still don't see any questions, and I don't think there are any hands up, but thank you very much,
There
Dr.
are
Kwong.
two questions
Oh,
in the chat, in the Q&A.
excellent.
Yep.
Okay, let's see.
Thanks,
So we have, since there are licensed RSV vaccines for adults, what is your suggestion for structure-based design for RSV vaccines for children?
So if the children have pre-existing immunity, then many of the vaccines that you're trying to increase the immunity might work just fine in the way they do for adults. However, if you're going earlier, it depends if they've taken an antibody, for example, and never been infected. And so your first course is going forwards. I do think that the stabilized molecules are the things to do that because they show the higher immunogenicity overall. And now the monkey and the mouse immunogenicity studies in naive animals might be an excellent way to predict what works there. And there, the disulfide stabilization does appear to increase the immunogenicity fourfold up and be really quite good. So my prediction for that is additional disulfide, really good disulfide molecules that are very, very stable and present the right conformation to the immune systems would be probably optimal for the childhood vaccines, pediatric vaccines. And the second question?
The second question, thoughts on enough homology of class 1 fusion proteins for broad antibodies or fusion inhibitors, even cross-viral class?
Well, that's a really interesting question. And the issue is that class 1 fusion proteins, they show almost no sequence homology going forwards. And so there are very great differences going forwards when you have no sequence identity all going forwards. There are different types of antibodies, however, that do cross really, really diverse things. And for example, we now have a glycan antibody that we're just identifying and characterizing that binds the glycans on HIV as well as the glycans on influenza. It binds, I think, RSV as well, but doesn't neutralize RSV. So there's a possibility that certain characteristics might go all the way out, but I think it's going to be very difficult to get high neutralization there going forwards, except in very particular cases, like in the case I just told you about, the glycan-reactive antibodies neutralize HIV quite broadly, over 50%. And then if you take the H3 strain of influenza, where the glycans have built up, once the glycans build up a certain density, now the antibodies start recognizing. But in terms of having antibody-based design going all the way out for everything, I don't think that's going to work so well. I think you're going to have immunogen-specific immunogens that are specific to each pathogen, and you're not going to be able to spread one pathogen over to many going forwards, but you have to teach the immune system for each one going forwards.
Excellent. And we have another question. Have you considered stabilizing and focusing on expressing only the most neutralizing epitopes for RSVPreF rather than stabilizing the entire protein?
Yeah, so one of the issues is, could you enhance this in other ways? For example, make a bunch of scaffolds or other things to just get the very, very best neutralization or not. And the problem is that if you make all these epitope scaffolds to try to do that, the real difficulty is that there's so much other recognition and lots of other surfaces that come. And so the question is, what's going to be most efficient ultimately going forwards? All the scaffold technologies that work you can show that they work, but they often have to have much, much lower immunogenicity. So I'd love to see it work. I think it's difficult. And ultimately, if you're presenting the whole target molecule for going forwards, I think that's ultimately the best thing to do with the stabilized trumps in this particular way. However, if you have pathogens that change sequence enormously, instead of recognizing the entire surface, you might want to recognize only on the conserved parts. And in those very particular cases with other pathogens with high sequence variation, that's how you can actually focus on particular sites of vulnerability. But I don't think you need that for RSV because, in general, the sequence variation of RSV is not so great.
We have several other questions. Okay. So we have one on talking about the cause of vaccines-enhanced respiratory disease about mRNA 1345 in children.
Sorry, can you repeat that? I missed that.
I think the person is asking if you can say anything about the potential cause of the vaccine-enhanced respiratory disease that was seen in the mRNA 1345 clinical trial in the pediatric population.
So as a structure-based designer, that question is probably better for Luis, but the issue is, as far Barney Graham explained this to me, is that if you get antibodies recognized in the wrong shape, and they're not very neutralizing, you can start having deleterious effects there going forward. And so you can have antibody enhancement in different ways if you have the wrong type of antigenic recognition. And so the post-fusion form appears to do that. So if you teach things to recognize the post-fusion, you get a lot more antibodies that are not neutralizing that can lead to other issues. But really, I'm not the expert on that. I'm really the one that says, and I think this is an important concept, if you're looking for a particular vaccine immunogen, please make sure that the antibody that you're using is really the form that you want. And if you do have something that doesn't neutralize or recognize other conformations, you have to validate experimentally what's useful versus not. And I think that's totally experimental. You don't know necessarily what's going to ultimately work. And in fact, it could be that HMPV, when we did it, we're agnostic to pre versus post. And so we actually stabilized both forms. And the post-fusion form gave, when stabilized disulfides, really high immunogenicity. So will ultimately work. It's a medical question which has all these different factors, and I think you just have to do that experimentally. That's what clinical medicine is going to be for.
Thank you very much. Excellent. And I think the other questions we had were different iterations of the same question you just responded to. But thank you, everyone, for the wonderful questions. And thank you, Dr. Kwong, for the excellent presentation and insightful responses. So we're going to go on to the next speaker. We have Dr. Hing. She's currently the Chief Scientific Officer at CynVac/Blue Lake Bio. And she's made a lot of contributions. She's contributed towards taking a lot of vaccines from bench to the clinic. So over to you, Dr. Jen.
Okay, I think you can see my slides, right? Let me just... Okay, thank you for the introduction. And here I'm going to present a PIV5 vectored RSV intranasal vaccine. We call it BLB201. And I will share some data on the safety and the immunogenicity of this vaccine in healthy adults and children. So this is the RSV vaccine monoclonal antibody snapshot, as mentioned earlier. We have a great success to get the protein-based the vaccine and the mRNA-based vaccine approval for adults. And the fusion protein-based monoclonal antibody and the Survimab got approval, which works really nicely to protect children less than 8 months old. However, the vaccine development in children has been extremely challenging. And recently, Moderna shared their mRNA vaccine caused the vaccine-associated enhanced respiratory diseases in December's World Health meeting. So the live attenuated or chimeric the vaccine viruses. So the live vaccine virus, there are 2 types. One is the live attenuated RSV vaccine developed by Codagenix, the NH group, under phase 1 clinical trial, and the leading candidate joined developed by Sanofi and NH in the phase 3 clinical trial. So the Blue Lake biotechnology, we have vaccine for older adults in phase 1 trial, which is completed, and the phase 2 trial in children. So what is the PIV5? It is the parainfluenza virus. It's a negative strand RNA virus like RSV. And this is a very ideal live replicating vaccine vector. So it is a component of the kennel cough vaccine. So original thought that this is the pathogen for dog, but later on, the research found that actually PIV5 does not cause kennel cough, and this is not a human pathogen. So the kennel cough vaccine containing the PIV5 has been used in dogs for more than 50 years. It has shown to be effective as the intranasal vaccine, and it has a proven safety record because dogs can share the PIV5 for up to 5 days, but there's no disease or infection reported through the dog owners or veterinary staff from exposure to PLV5. And the PLV5 can elicit very good immunogenicity, not only the seroantibody, mucosal IgA, and also very good cellular cell-mediated immune responses. The virus is very stable. We have formulation on study. At the 40°, it can have stability more than 4 months, and at -20°, more than 2 years stability. The virus grows really high yield, and so it can grow in zero-free virus cells, very high titer, up to 9 logs per ml, and require minimal downstream purification. So it provides a very cost-effective vaccine platform. And okay, I show you this is the dog receiving the intranasal vaccine. So, all right. And we have developed the PLV5 based vaccine and have 2 clinical stage program. The number 1 is the SARS-CoV-2. We finished the phase 1 trial in 72 adults, and followed by a phase 2A study, including the elderly population. So 227 people were enrolled in the COVID outbreak season, and through this study, we have preliminary data show that the vaccine offered protection against the symptomatic COVID infection in the first month. And currently, we have a phase 2B clinical trial still active, which is sponsored by BARDA HHS. The plan is to enroll 10,000 people. We have enrolled more than 400 participants already. And RSV vaccine, the phase 1, we complete the enrollment in 33 to 75 years old. The paper has been published. And currently, in children 8 to 59 months old, we have enrolled 63 participants. So with more than 400 people receive the vaccine doses in ranging the age range 8 months to 80 years old in US, we have shown that the vaccine is extremely safe. There's no safety concern from the people received our intranasal vaccine. So what is the BLB201? This is the vector, the PLV5 vector in yellow with the Y-type RSVF protein gene inserted between the SH and the HN gene junction. So we decided to use the F protein, which is exactly the same as found in the Y-type RSVF, other than the prefusion stabilized F protein used for the protein vaccine or mRNA vaccine. So Dr. Biao-He's lab has published many papers to show that PLV5-based RSV vaccine is safe and efficacious in mice, cotton rats, and non-human primates. As you can see here, the BLB201 infected viral cells express both PLV5 and protein and RSVF antigen. So this is an initial study conducted in mice, showing we have compared the PLV5 expressing the native F and the pre-F form. So in mice model, you can see that both native F and the pre-F elicit high titer of pre-fusion form F IgG antibody, and also the post-fusion F IgG, which is higher in the native F than the pre-F form. However, if you look at the neutralizing antibody titers, the total F, the native F, has higher neutralizing antibodies compared to the pre-F form, which is the difference is about 30-fold. So that's the reason why we choose the RSV native F as the RSV vaccine candidate. And this is, again, the mice study compare PLV5 expressing the native F versus RSV A2 strain, the F IgG antibody, and the RSV nude antibody. So you can see the titer is, the IgG titer is slightly lower than RSV A2, but it provided 100% protection in the mice challenged with RSV A2 virus. And as you all know, that the formerly inactive RSV vaccine used in 1960 caused enhanced disease, which is represented by interstitial and alveolar inflammation. And you can see this is the cotton rice model with associated disease. So it has high score. However, with the BRB 201, you can see we use interstitial and alveolar inflammation, and the low dose has inflammation similar to the RSV A2 infected animal control. So this data really show that we did see the low dose breakthrough infections and did not see the vaccine enhanced disease signal in this cotton rat model. And then we did the primate study to look at the Th1 and the Th2 type immune response. As you can see that the BRB201 elicits a very good CD4 positive and CD8 positive cellular immune response, which is specific to the stimulation with RSVPreF peptide, not with the COVID S antigen expressed by PLV5. And looking at the cytokine level, the type 2 cytokine IL-4 and IL-5, IL-13, they are very low. So based on the promised preclinical study, we went ahead and did the phase 1 clinical trial in healthy adults, total enrolled 30 people, half young adults and half older adults. So we can see the vaccine virus has the shedding can be detected in 21 or 13% trial population. And in this trial, we did not pre-screen the adults for the pre-existing RSV antibody titer. So we have very high baseline RSV antibody detected in those participants, but we were able to see the increase of neutralizing antibody on day 15 and the day 29 compared to the pre-vaccinated level. This is control group. The blue one is the vaccine group. And we also can see the PLV5 vector IgG antibody response in the vaccine group. This is elderly. Sorry, the blue one is young adults, and then the orange color is the elder one. So we can see the PLV5 antibody response in both population. So this trial to tell us that BRB201 is safe and well tolerated, very minimal reactogenicity. And this phase 1 study, we also did the CMR assay, and showing the young adults and the older adults, we can detect the CD4, CD8 positive T cells post-immunization. And we had the IL-13 included in the cytokine panel, and we did not detect any signal from any cells secreting IL-13 cytokine. So that indicates the CMI response is not Th2 biased. So based on the adult trial, we show the safety and immunogenicity in seropositive people, and we then proceed with pediatric study. This is a phase 1 and 2A study. The primary objective is the safety to healthy 6 to 59 months old children, and the secondary is immune response in serum and nasal secretions. We also have exploratory endpoints to examine the frequency of symptomatic ISV infections in the trial population. So the study design is, we were really careful to use sentinel and the secretion cohorts to do vaccine dose escalation, to start with the lower dose and higher age, and go down with higher dose and younger age groups. So the DSMB review after each group sent in the cohorts, and also the enrolled after each group were enrolled. We have safety post criteria and 1-year follow-up for the MAAE, SAE, ISV infections, and the diagnosis of any lower respiratory tract infection. So this is the phase 2A pediatric study enrollment status. We have completed the group 1 and 2 seropositive children, 18 to 59 months old, at the dose level, lower dose 6 logs and the higher dose 7 logs. Those are all seropositive, and half vaccine group and half placebo group. So the group 3 is the lower dose in the younger children, 8 to 24 months, and including seropositive and seronegative children. The last patient has finished the last visit, and we have a total 18 doses. So this group is completed. And we were actively enrolling the higher dose group in this group 4, single dose, and group 6, 2 doses. It was started last year. However, due to the Moderna vaccine enhanced disease, FDA put the seronegative children trial enrollment on the pause. So here's a summary with the 63 children we have enrolled. In terms of safety, we did not observe any vaccine-related SAEs or MAAEs. The vaccine replication mostly detected in the high-dose group and very little in the low-dose group. We had a total of 8 ISV cases reported. None of them severe, very mild symptoms, and there's no vaccine-associated enhanced disease. So to prepare for the last December's WPAC meeting, we unblinded the ISV infection data, and then we showed that there's a statistically significant vaccine efficacy against symptomatic ISV infection at the VE rate more than 80%. And because of the group 1 and the group data have completed, the enrollment completed, and we unblinded the immunogenicity data. And then we show you here with the 7 logs PFU group 2 group, we can detect the ISV nude antibody. Left is the vaccine group, and the right, the orange color, is the placebo group. So we see the more than 8-fold increase in ISV neutralizing antibody. And then we see the ISV F0 IgA, F IgA increase. Also, PLV5 neutralizing antibody increase. Most importantly, this here is we can also detect ISVF specific nasal IgA increase by more than twofold. So this is the summary of the BLB RSV vaccine clinical study. Seropositive adults at the dose level 7.5 log PFU, we see the safety. Overall, 17% replication. We detected RSV antibody response in 64% trial participants, and then PRV5 neutralizing antibody response 59%, cellular immune response 97%. In seropositive children, this is the group 2. I just showed you earlier the safety replication 60% participants. RSV antibody response about 80%, and the PRV5 neutralizing antibody response 80%, which about the same. And we only had 2 vaccinated subjects tested for CMI response. We can see 100% cellular immune response. For the mixed population, seropositive, seronegative children, 6 to 24 months, given 7 logs, we show the safety replication, and we can detect the RSV antibody, PRV5 antibody response. And however, the trial still blinded. We don't have the final data to report. So the conclusion, we showed here that BLB201 intranasal vaccine expressing Y-type F protein does not produce type 2 biased response in animals or humans. BLB201 is safe and immunogenic in both RSV seropositive adults and seropositive, seronegative children. 63 children have been enrolled without evidence of vaccine-enhanced disease. PRV5 vectored intranasal vaccines are most likely protective based on preliminary analysis showing vaccine efficacy in 8 to 59 months old of 80% in preventing symptomatic RSV infection. So we believe that this vaccine is a very promising RSV vaccine for further clinical development. So this is acknowledgement. The Blue Lake and the CynVac, Christina and Christine are the 2 leading scientists that work on this program. And the funding, Biao He is the founder of the company, and he has spent at least 2 or 3 decades on the PRV5 vector for the vaccine development. So University, Louisiana, and Lafayette, Penn, and Franz Schwab's, they did the CMI response for us, and we have a clinical trial principal investigate from CSHMC, Paul Spearman. The clinical sites is CSHMC and the Baylor College of Medicine and some commercial sites. And especially, I'd like to thank the trial participants and especially parents for allowing their children to participate in the trial. And thanks for your attention.
Excellent. Thank you very much, Dr. Jane. Thank you for the presentation. We have some questions in the Q&A. So I think the first question, you already addressed it during your presentation. It was asking if you have any clinical trials, including infants younger than 8 months, especially 5 to 7 months, right? But I think you showed us.
Yeah. We originally planned to include the infants 6 months and older, but because the nirsevimab was approved for infants less than 8 months old, so we decided to only address enrolled children more than 8 months old.
And I think before we go on to the next question in the Q&A, I have a question following up on that question, because I think it's trying to get at how much exposure have you had to seronaive children? So do you have any thoughts on the reliability of the preclinical models for vaccine-enhanced respiratory disease, right? Because historically, we talk about cotton rat model, we talk about primate model, and we know that with recent evolution, it doesn't seem like we can generally say that if you don't see any signs of vaccine-enhanced respiratory disease in those models, then you're fine to go into the right seronegative children. So any thoughts on any additional preclinical models to assess that?
Yeah, I think the preclinical model can provide valuable data to guide us. And we actually, because of mRNA, mRNA vaccine, Moderna did a very good job conducting cotton rat study, and they did not see the vaccine has disease, but they did have an issue in the infants trial. So the FDA required us to conduct another comprehensive cotton rat study. So we're working with the FDA to try to resolve the partial clinical hold issue. For our vaccine, we have confidence we don't have the vaccine enhanced disease issue because we use live virus and natural RSVF protein, which is present in RSV virus, and RSV infection never cause enhanced disease. And the same as the live attenuated RSV vaccine, which is being carried on by the NIAID group for many decades, and the current Sanofi's trial. So I don't think a live vaccine would have an issue causing vaccine-enhanced disease. And how the cotton rat model, I know preclinical model may not be 100% reliable. Otherwise, we don't need to do the clinical trial, right? So we have confidence our vaccine is safe.
Excellent, thank you. And we have another question here. So it says, since this is a vector-based vaccine, do you have any data on whether it can be used multiple times without impacting immunogenicity?
Yeah, this is an excellent question. It's always a question about the vector immunity, right? When you have a seropositive individual, whether the vector still works? And the answer is yes, because we did the dog study, immunized PRV5 first, and gave PRV5 vector vaccine. We see a very good immune response. And in the clinical study, we see PRV5 positive participants ranging from 10% in children to 30% in adults. And their positive status does not prevent the antibody response from immunization. And also, if you look at the paper published by Biao He, in dogs, I believe, the PRV5 antibody titer very low in the serum. It's less than 100 or the 60 on average. So somehow the PRV5 does not elicit a very high serum antibody. But we need more clinical data to prove it. The current finding is this is not an issue for us.
Thank you. And final question we have in the Q&A, can you comment on your further clinical development plans?
So yeah, we would like to continue finish the phase 2A clinical trial, and then start the phase 2B or phase 3 trial if possible, but one stage at a time. So we keep, we're working with FDA to let us to go to complete the phase 2A first. Yeah.
Excellent. Well, thank you. We'll be on the lookout for what comes next with you. Thank you very much for the presentation. I'm going to go over to our final speaker for today, Dr. Kumar Ilangovan. He is the Director of Vaccines Clinical Research and Global Clinical Lead for the RSV Adult Vaccine Program at Pfizer, and is going to talk to us on the PreF vaccine in older adults and at-risk adults aged 18 to 59.
Thank you so much, Dr. Chime. Good morning, good afternoon, good evening, everyone. Thank you so much to ISV for the opportunity to present. My name is Kumar Ilangovan, and as Dr. Chime said, I'm the RSV Adult Global Clinical Lead at Pfizer. I'm very pleased to present clinical data for the RSVPreF vaccine in older adults, younger at-risk adults, and the immunocompromised adult population. Here are my disclosures. Abrysvo, or RSVPreF as it's referred to in the clinical trials, is a bivalent vaccine composed of engineered, stabilized, trimeric Prefusion-F glycoproteins matching the two RSV subgroups A and B to help ensure the broadest coverage against RSV illness. The current indications for Abrysvo include active immunization of pregnant individuals at 32 through 36 weeks gestational age for the prevention of lower respiratory tract disease and severe LRTD caused by respiratory syncytial virus in infants from birth through 6 months of age, and active immunization for the prevention of LRTD caused by RSV in individuals 18 through 59 years of age who are at increased risk, and active immunization for the prevention of LRTD caused by RSV in individuals 60 years of age and older. Before providing an overview of the adult clinical trial results, I wanted to share an overview of the maternal indication and status of approvals. As of April 2025, 64 countries have approved RSVPreF vaccine for use in pregnant individuals and adults, including the 30 countries within the European Economic Area. In addition, RSVPreF is also approved for use in Macau and Hong Kong special administrative regions. RSV vaccination is recommended by multiple national and regional bodies for the pregnant individuals to help protect infants. On this slide are the descriptions and various gestational ages for which the vaccine is recommended. In September 2025, SAGE discussed RSV in young infants and the potential role for passive immunization in preventing RSV-related disease based on real-world RSV immunization programs. The recommendations were posted in December 2024, and their final position paper is forthcoming. For maternal vaccination, SAGE recommended a single vaccine dose in the 3rd trimester of pregnancy as defined in the local context. A year-round approach to RSV vaccination is preferable in most countries in tropical and subtropical regions where RSV circulates for much of the calendar year, or where seasonality patterns are not well described. Subsequently, in March 2025, the WHO pre-qualified Abrysvo to protect infants against RSV-associated disease during the first 6 months through the transfer of antibodies during pregnancy. In November 2023, the PAHO Technical Advisory Group on Immunization held its 11th ad hoc virtual meeting to consider the safety and efficacy of the maternal RSV vaccine for the prevention of RSV-associated disease in infants, among other topics, and published their recommendations in January 2024. For countries or territories of Latin American or the Caribbean that elect to introduce the maternal RSVPreF vaccine, PAHO-TAG recommends the use of the RSVPreF vaccine in pregnant women at 32 to 36 weeks of gestation to prevent RSV disease in infants while minimizing the risk of preterm birth. Between October 2023 to April 2025, more than 1.3 million doses of Abrysvo have been administered to pregnant individuals in select countries with maternal vaccination programs. Now I'd like to move on to a summary of the RSV Adult Clinical Development Program, which has included the pivotal efficacy study, Renoir, on the left here, conducted in adults 60 years of age and older, which continues to study revaccination at 1 through 4 year intervals from initial vaccination, and the Monet study in adults at high risk of severe RSV disease. The clinical development program has also included 2 completed studies of RSVPreF co-administration, the first with COVID-19 vaccine with and without high-dose influenza vaccine, and co-administration with adjuvanted influenza vaccine, both of which met criteria for non-inferiority of co-administration. Lastly, after the approval of Abrysvo, multiple post-authorization studies began to better understand and monitor the vaccine's safety and effectiveness. I would like to begin by sharing the results of the Renoir pivotal efficacy study. Renoir was the global study in over 38,000 participants 60 years of age and over that demonstrated safety and efficacy through 2 RSV seasons. As you see in turquoise, surveillance in seasons 1 and 2 was for an average of over 7 months each, for a combined surveillance period that averaged over 16 months through the 2 RSV seasons. The Renoir data demonstrate that efficacy against lower respiratory tract disease due to RSV remained high through two RSV seasons following a single dose of RSVPreF. Efficacy in season 1 was 88.9%, shown in dark blue, and efficacy in season 2 alone remained strong at 77.8% in light blue, for a combined efficacy across 2 seasons of 81.5% in green. A subset of participants in the US and Japan had blood samples collected before vaccination, 1 month post-vaccination, and at the pre-season 2 time point. RSV neutralizing geometric mean titer responses for RSV A and B subgroups were robust 1 month after initial vaccination and remained nearly 5-fold elevated above baseline titers at the pre-season 2 time point. The Renoir study was subsequently expanded to study the safety and immunogenicity of RSVPreF revaccination at 1, 2, 3, and 4-year intervals from initial vaccination. Participants from the main efficacy study who received RSVPreF were enrolled in these revaccination studies and will either receive RSVPreF at the specified interval or placebo and will be followed for safety and immunogenicity assessments to further describe the immune responses and safety after revaccination, as well as immune kinetics of a single dose. Co-administration of RSVPreF was also studied in the 2 separate trials mentioned earlier. One in which RSVPreF was administered with a COVID-19 vaccine with or without high-dose influenza vaccine, and a second trial where RSVPreF was administered with adjuvanted influenza vaccine. Responses in these trials were compared to standalone vaccine administration. In the study examining the safety and immunogenicity of RSVPreF vaccine administered with adjuvanted seasonal influenza vaccine, all 4 HAI titers and both RSV neutralizing titer geometric mean ratios met the pre-specified success criteria of a lower bound confidence interval greater than 0.67. The study conducted to assess the safety, tolerability, and immunogenicity of co-administration of RSVPreF with COVID-19 vaccine and influenza vaccine was designed to show a 2-fold non-inferiority or a margin above 0.5. Among the participants who received RSVPreF and COVID vaccine co-administration, the geometric mean titer ratio met the pre-specified 2-fold non-inferiority per protocol and met a more stringent 1.5-fold non-inferiority for all 4 antigens shown by the dotted line. Participants who received RSV, COVID, and flu vaccine co-administration demonstrated a geometric mean titer ratio that met the 2-fold non-inferiority per protocol shown by the dotted line. The Monet study consisted of 2 sub-studies. I'll begin by presenting the results of the first, which was a double-blind study of RSVPreF in adults 18 through 59 years of age at high risk of severe RSV disease. The results of the study were the basis for the US approval of Abrysvo for the 18 to 59 high-risk indication. Our application for licensure in adults 18 to 59 years was driven not only by the successful Monet study, but also by the acceptable safety profile demonstrating over 5,400 adults aged 18 through 59 years of age who from our maternal and adult clinical studies. Regarding the immunogenicity endpoints, the success criterion was met by 4 co-primary endpoints. An analysis of covariance model was used to adjust for participant gender and baseline titers. The first 2 endpoints, shown in the top figure, evaluate the neutralizing titers 1 month following vaccination in adults 18 to 59 years with comorbidities and a ratio against samples from those 60 years and older from the prior pivotal Renoir study. The geometric mean titer ratio, or GMR endpoints, met the 1.5-fold non-inferiority margin for RSV A and B with lower bounds well above the 0.667 value shown with the dotted line. Similarly, seroresponse endpoints were also met for RSV A and B as shown by the bottom figure, with lower bounds well above a negative 10% difference. The second of the Monet substudies was a single-arm, open-label, multi-center study of RSVPreF among immunocompromised adults 18 years of age and older. The study assessed the safety, tolerability, and immunogenicity of 2 doses of RSVPreF 1 month apart. This graph shows immunogenicity results before dose 1 in dark blue, after dose 1 in the lighter color blue, and after dose 2 in the lightest color blue. In this immunocompromised population for both RSV A and B, immune responses after dose 1 were robust, demonstrating an 8 to 9-fold rise above baseline titers. Geometric mean titers did not increase further after a second dose 1 month after the first dose. Similarly, the geometric mean fold rises, GMFRs, shown across the bottom were highest after the first dose, with no increase after a second dose 1 month later. In this subgroup analysis by immunocompromised group, RSV-AB GMTs and GMFRs demonstrated that all groups, including solid organ transplant, autoimmune inflammatory disorders, non-small cell lung cancer, and end-stage renal disease on hemodialysis, had robust responses 1 month post doses 1 and 2. Interestingly, participants with end-stage renal disease on hemodialysis had the highest baseline and post-dose responses in the trial. When comparing RSV GMTs and GMFRs across studies, it was observed that the responses we see in the Monet immunocompromised and high-risk population, highlighted by the green box, were similar to the responses observed in the Renoir older adult study on the left. On the right are the results of the study in younger adults with chronic conditions. Thus, all ages and at-risk groups demonstrate high titers comparable to those seen in a study demonstrating efficacy for at least 2 RSV seasons. In the absence of a correlative protection, the results suggest a robust neutralizing titer response is achievable in the immunocompromised population after a single dose, and fold rises are likely high enough to confer protection. The clinical development plan presented today describes the results of Abrysvo in adults aged 18 years of age and older, including those with underlying condition, such as chronic or immunocompromised conditions, as well as the elderly. The Renoir study showed that duration of protection is at least 2 years with high efficacy in season 2, and neutralizing titers are robust against RSV A and B. RSVPreF was also shown to be safe, well-tolerated, and showed non-inferior immune responses when co-administered with influenza, COVID-19 vaccine, or both vaccines together. RSVPreF given to adults 18 through 59 years of age in the Monet study demonstrated robust RSV A and RSV B subgroup neutralizing responses that met non-inferiority to the Renoir study, where safety and efficacy were demonstrated. In the Monet immunocompromised adult study, safety and immunogenicity were demonstrated for a single dose of RSVPreF in immunocompromised and high-risk adults. Thus, RSVPreF addresses a substantial burden in older adults and those with chronic conditions. Prior to concluding, I wanted to share important results of the real-world studies of vaccine effectiveness of the RSV vaccine. Here are the available real-world data studies assessing RSV vaccine effectiveness in older adults. All studies are from the US due to its earlier launch. Here are the results of the RSV-associated vaccine effectiveness against hospitalization or emergency department hospitalization in adults greater than or equal to 60 years of age. All studies except for the one at the Kaiser Permanente Southern California Hospital included both RSVPreF and RSVPreF3. You can see that the values for overall population range from 75 to 89% and 72 to 73% for immunocompromised persons in terms of vaccine effectiveness. Here are the RSVPreF specific estimates from these studies ranging from 73% to 89% vaccine effectiveness. Abrysvo was approved in November 2023 in the UK, and shortly following this approval, entered into the UK national recommendations for use in adults 75 to 79 years of age. Abrysvo was the only RSV vaccine introduced in the UK and was subsequently launched in Scotland in August 2024 and England in September 2024. Vaccine uptake in both Scotland and England was high, 68% and 47% respectively. In Scotland, the pre-vaccination period in 2023 is shown on the left here, and the post-vaccination in 2024 on the right here. As you can see when comparing both years, the chart on the right shows a 62.1% reduction in the RSV-related hospitalization incidence in the target population. In England, on the left, we see the RSV vaccine coverage over time, with the start of the study period marked by the red line. On the right, we see a reduction of 30% in RSV-related hospitalization incidence nationwide. These real-world results indicate that in this targeted population, Abrysvo was effective in reducing RSV-related hospitalization rates. Thank you so much for your time. I'm happy to answer any questions.
Excellent. Thank you very much, Dr. Ilangovan. Wonderful presentation. I don't see any questions in the Q&A. Do we have any questions from the audience? I don't see anyone's hands up yet. Nothing yet. Wonderful. Can you tell us more about the future plans for the clinical development, RSV older adult clinical development program at Pfizer?
Absolutely, sure, happy to. So as shared earlier, we do have a study that is ongoing, furthering the research we're doing on co-administration, which is looking at another common co-administration that's observed, which is with the shingles vaccine, in this case, Shingrix. And that study has started and is actively enrolling participants 50 years of age and older. Regarding the post-authorization safety and effectiveness studies, we have multiple safety studies that are ongoing, as well as a real-world effectiveness study, which was mentioned earlier, the Kaiser Permanente system, in adults 60 years of age and older. This study in particular has enrolled, is an observational study where Abrysvo is being administered in this population, and the population has a breadth of representation that's greater than was observed in the Renoir trial, meaning that there's more older adults and those with comorbid conditions, and including immunocompromised and high-risk populations, to further understand the vaccine effectiveness in the real-world setting. This real-world effectiveness study has shared data for the first season and is planned to continue for 5 RSV seasons.
Thank you. And with the Shingrix cohort study, is it you have adults greater than or equals 50 years of age? Are they healthy adults, or are you just looking at adults at increased risk or immunocompromised?
Great question. It's among healthy adults and those with chronic stable conditions, similar to how we enrolled the Renoir study.
Okay, great. Excellent. Well, thank you. I think we have some questions. Great. So how do you explain that there appears to be no effect? Oops, I think the question disappeared. Sorry, whoever typed that question, can you please retype the question? But I'll go on. Yeah.
Appears to be no effect in the population over 80 years. It's a great question. So the reason for this lack of effect in the population above 80 years is that the recommendation in the UK was only made for adults 75 to 79 years of age. There is no population that is recommended to receive the vaccine after 79 years of age. So the results of this study are quite indicative of the effect within a targeted population. Hope that answers the question.
Well, sounds like it did. But with your phase 3 trial, you had adults older than 80 years of age, right, enrolled in that clinical trial. And did you see efficacy? Yes,
we did. So within the Renoir clinical trial, just going back to the vaccine efficacy, so even in subgroup analyses, when looking by age, the efficacy that we observed for the overall population was quite similar. In the 80 years and older age group, due to lower enrollment than compared to the younger age groups within the study, the efficacy was 100%, but the confidence intervals were quite wide due to the lower number of cases.
But at least we can say there's a signal of efficacy in that age group.
Yes.
Wonderful. So the next question, please comment on UK recommendation, age recommendation, and vaccination coverage in relation to the study results presented or published.
Yes, it's a great question. So the JCVI and UKHSA have made the recommendation to cover the 75 to 79-year-old population for the first season. And I believe that the policymaking bodies in the UK are continuing to evaluate the recommendation. So I don't have any further updates, but that is the current status. It's recommended as a single dose for 75 to 79-year-olds in the UK.
And we have a question on the vaccine, the maternal vaccine. So how long did the vaccine protection of the baby from the mom last?
Great question. So the vaccine efficacy within the Matisse clinical trial was studied through 6 months after birth. So the protection and efficacy estimates within that clinical trial are for 6 months post-birth, and similarly reflected in the indication, which states for birth through 6 months of age.
Great. I think we have addressed all the questions. Do we have any additional questions from members of the audience? I don't think so. Excellent. Well, thank you. Thank you very much, Dr. Ilangovan, and thank you to all our presenters today. And thank you to everyone for making out time to attend this mini symposium. The next one is tentatively scheduled for the end of June and will focus on HIV. Well, thanks, everyone.